Okay, let's move to the next talk. So please welcome Dr. Yifei Li from Zhejiang University.
Thanks for coming today. It's really nice to be here, and today I'm going to talk about our ongoing project about glymphatic clearance and its association with network connectivity in human. Okay, I have no disclosures. As we all know, neuronal activity inevitably produces waste. Billions of neurons with high metabolic turnover rates and spatially elaborate layouts are packed into the brain. But how the brain extraordinary activity coordinates its complex clearance system, especially notably in cortex, neural activity is regionally synchronized across microscopic networks. For now, we have no clear picture of how the clearance how the cortex modulates its clear. Sorry, how to turn over to the previous slides? Oh, okay. But for now, we have no clear picture of how the cortex has its clearance pattern, so let alone the networks. So a very natural question comes to us: can we picture this, and does network coordinate its clearance with its structural or functional properties? Before my work, I want to give you some brief introductions. What is glymphatic system? The recent discovery of glymphatic system offers a novel perspective to understand brain waste clearance process. In this recirculation system, cerebral spinal fluid enters the brain through perivascular spaces and enters into the interstitial compartment, finally drains out metabolic waste, including A beta and tau, to the meningeal lymphatics. It is only until recently, maybe months ago, 3 teams independently reported that neuronal dynamics direct cerebrospinal perfusion and brain clearance. It is thus worthwhile to comprehensively visualize and assess network glymphatic clearance in human. So how can we assess glymphatic clearance? The gold standard is called glymphatic MRI. It is very easy by taking serial T1 images before and at multiple time points after intrathecal administration of contrast agent. We can easily calculate the signal percentage change at each point from each ROI. Meanwhile, non-invasive regional assessment for glymphatic clearance is lacking. Previously, some methodologies have been proposed, like DTI-ELF index, but this index also assess whole brain glymphatic clearance as average. Hindered many further investigations, so here we propose that free water might be a potential candidate. By using tensor model, free water is defined as the water molecules that do not experience flow along the wet matter bundles. And thus, it could be theoretically be assumed as an interstitial element of parenchyma, an important part of our glymphatic system. So this study mainly included 2 cohorts. In participants who underwent glymphatic MRI, we tried to identify the network-based glymphatic characteristics and to validate the relationship between non-invasive free water and glymphatic clearance function. Then, in extended community dwelling cohort as registered in clinical trials, we tried to understand whether network-specific glymphatic clearance function, as indicated by non-invasive free water index, coupled with network connectivity properties, both in cross-sectional and longitudinal studies. So, in patients admitted to our hospital with indications for lumbar puncture and volunteers in this study, finally, 85 patients underwent glymphatic MRI were finally included. Based on Schaefer 400 puzzle 7 network atlas, we calculated glymphatic dynamics for each regions And we define two indexes for network level glymphatic clearance. That is the mean of each network, reflecting the mean clearance function of each network, and the standard deviation inside each network. The signal percentage change was calculated as below. And yes, regional glymphatic dynamics across cortex is spatial-temporally non-uniform. You can see that the signal percentage change from 4.5 hours increases to 15 hours, then to a decrease in 39 hours. So the 39 hour is the clearance phase, and we thus assume 39-hour percentage change to reflect the glymphatic clearance function. As you can see, the highest 39-hour percentage change located mainly in the posterior cingulate cortex, precuneus, dorsal-medial prefrontal cortex, and the parietal cortex. Consistent with our level results, the network also peaked at 15 hours, then to a decrease in 39 hours, further confirming the 39-hour is the clearance phase. Glymphatic clearance characteristics do vary among functional networks. The whole picture is presented at figure A, and in B, the color and value represent those mean glymphatic clearance of each network in x-axis minus those in y-axis. You can see that limbic network was the one with the lowest percentage change, while the dorsal attention and default network was of the highest. This is pretty interesting since in some pathological conditions like A-beta accumulation, it follows a spatial trajectory. You may know that as A-beta, it spreads predominantly from default network, especially in precuneus or posterior cingulate cortex, then to a set of lower order areas. And for confounders, we only find age and subjective sleep quality generally correlated with all network mean glymphatic clearance. And all those have survived adjustment for demographic confounders and FDR correction. Age is also correlated with 39-hour percentage change across 335 regions after FDR correction. For heterogeneity, default mode has the greatest heterogeneity, and the limbic has the highest, has the lowest, and the default mode has the highest. And age only age was positively correlated with all network heterogeneity, and it's also survived adjustment. Then in patients with valid DTI images, we further applied bi-tensor model to map free water, try to validate free water with glymphatic clearance both in RI and the network level. The whole picture of free water was demonstrated on figure A, and across 400 puzzles, 39-hour percentage change was positively correlated with free water value. And 200 and in group level, 223 regions passed FDR correction and adjustment for potential confounders. Consistent with previous results, network-free water was the lowest in limbic network and the highest in dorsal attention network. Almost all networks passed FDR correction and adjustment for potential confounders except for somatomotor network, though with a tendency. Also, network-level free water heterogeneity was positively correlated with glymphatic clearance heterogeneity. Finally, in 287 community dwelling participants with valid baseline and follow-up multimodal MR scans, we constructed structural connectome and functional connectome based on DTI and resting state fMRI. It turns out that network-level free water was negatively associated with inter-network connectivity both structurally and functionally. Here the R represents univariate analysis, and in beta we adjusted for potential imaging confounders and demographic confounders. However, in functional connectivity analysis, the default mode and the limbic network do not pass the adjustment in baseline cross-sectional analysis. In longitudinal analysis, we found that the decrease of free water was negatively associated with improvement of structural net connectivity, and even after adjustment for baseline free water, follow-up years, and other demographic confounders. Though somatomotor network did not pass the adjustment, though with a tendency. And in functional network, only the change of functional network in dorsal attention network and somato network finally is negatively associated with changing free water index, and both networks passed further adjustment. So in summary, utilizing glymphatic MRI, we profiled a hitherto unrecognized glymphatic clearance pattern across cortical networks, with dorsal attention network and default mode network being the worst, while limbic the best. Free water as validated with glymphatic MRI could be a region or network-specific non-invasive measurement for glymphatic clearance. These studies underscore a necessity for glymphatic health in maintaining proper network integrity and function. Our findings may provide a new perspective to investigate network-based glymphatic clearance function, which particularly works well in neurological diseases with spatial clinical pathological features. I would like to give my acknowledgement to my mentor, Minlo, in Zhejiang University, and my colleague Xiaozhu also contributes a lot to this project. Thanks for Professor Maridio in Barrow Neuroimaging Innovation Center for free water modeling. If you have any questions, please feel free to contact me or visit my poster.
You have time for a very quick question. There's a question online. How do you interpret your association between free water and functional FC and SC biologically?
Wow, it's a hard question. And we have considered, sorry.
Maybe just a quick answer. We do not have that time to explain.
Okay. Focusing on longitudinal analysis, we can see that the free water change is mostly associated with structural connectivity. It may be because that free water is a structural change, so that structural connectivity is more sensitive to this change. And for functional connectivity, it may be because that it is more flexible and may be and may have compensate mechanisms to solve this problem. And yes.
Okay, thank you.